Assalamualaikum and good day, students. This is a very short topic on antiglobulin test and its application. Antiglobulin test is also known as Coombs test. This test is widely used in transfusion testing, and we use anti-human globulin as a reagent. So, AHG reagents may be polyspecific and also monospecific. What is the difference between polyspecific and monospecific? Polyspecific AHG contains antibody to human IgG and C3d component of human complement. Monospecific AHG contains only one antibody specificity, either an anti-IgM or anti-IgG or specific component of human complement. So actually, AHG is an antibody against the human globulin. So how do we prepare this reagent? This reagent is prepared by injecting human serum or purified globulin into the lab animals such as rabbits. And when the animals receive this human serum, it is alien to the rabbit's body. So it behaves as foreign antigen towards the rabbit. So the rabbit immune response will be triggered, and an antibody to the human globulin is produced. So this is how we prepare AHG. First, we inject the human serum into the rabbits, and then the rabbit will produce antibody against the human serum or against the human globulin. Then we will extract the AHG from the rabbits, and then we will get either polyspecific serum, polyspecific reagent, or monospecific reagent. There are two types of antiglobulin tests. One is direct antiglobulin test or DAT. The other one is indirect antiglobulin test or IAT. We also usually call direct Coombs test DCT or indirect antiglobulin test ICT. For DAT, we want to detect whether the antibody or complement coating of red blood cell has occurred in vivo. In other words, we want to know whether a patient's red blood cell has been coated with an antibody or a complement that has occurred in the patient's circulation. So this is the principle of direct Coombs test or direct antiglobulin test. In direct Coombs test, we want to test whether the RBC of the patient has been coated by the antibody in the patient's body. So we will take blood from the patient, and then we will take the RBC from the patient. So if the patient's RBC has antibody coated on the red cells, then we will get this kind of thing, and then we will put the AHG to the solution. So this AHG will cause the red blood cells to agglutinate, because the AHG will link the RBC by binding to the human antibodies on the RBC. So it will cause the red blood cell to bind together. So this is another diagram to show you the positive direct Coombs test. So if the antibody has been coated the RBC of the patient in the patient circulation, when we put in the AHG, this AHG will link towards the antibody on the red cells of the patient, and this will cause the red blood cell to be agglutinate, and we will see the positive results. So for example, if the patient does not have any antibody on the red cell, so when we put in the AHG, we don't see any agglutination because there is no antibody on the red blood cells that can bind to the AHG. Okay, What can be the causes of positive DAT? DAT usually is positive when we suspect immune-mediated hemolytic anemia, or in other words, antibody-mediated destruction of red blood cells. So a positive Coombs test indicates that an immune mechanism is attacking the patient's own red blood cells. This mechanism could be autoimmunity, alloimmunity, or a drug-induced immune-mediated mechanism. In other example, we can also have a positive DAT in hemolytic disease of newborn and also hemolytic transfusion reaction. Okay, for indirect antiglobulin test, this is quite different from the direct antiglobulin test, in which IAT is we want to detect red cell antibodies that are free in the serum. For DAT, if you still remember, we want to detect whether the antibody has caught the RBC of the patient in the patient circulation. Okay, so in DAT, we use patient's red blood cell. But in IAT, or in indirect antiglobulin test, we use the patient serum because we want to detect whether the patient serum has the red cell antibodies. IAT is usually used to detect very low concentration of antibodies present in a patient's plasma or serum prior to a blood transfusion. So usually we use it in antibody screening, antibody identification, cross-matching, red blood cell phenotyping, and also titration studies. Generally, direct Coombs test has only one stage, because we take the patient red blood cell and we put in the AHG, and then we see whether the patient has positive or negative reaction. But the IAT has two-stage procedure. First, we will have to incubate the patient serum with the normal RBC. The normal RBC does not come from the patient. So first we incubate the patient serum to the normal RBC. So if the patient serum has the antibody, that antibody will absorb onto the red blood cell of the normal red blood cell. After that, we will wash whatever remaining antibodies. Then we will do the direct Coombs test, meaning that then we will put in the AHG and see whether the patient has agglutination or not. If the patient has agglutination, that means the patient's serum has antibody against red blood cell. But if the patient does not have, or if the patient have negative reaction, the patient does not have antibody against the red blood cell. Okay? So this is the principle of the IAT. First, we will take the patient serum and then we will incubate the patient serum with the normal RBC, or we call it reagent RBC. So if the patient serum has the antibody will bind to the RBC antigen, and then we will wash this solution, and then we will put in the AHG. The AHG will bind to the antibody on the red blood cell and cause the agglutination of the red cells, and then we will have a positive result. There are few methods that we can use for antiglobulin testing. The first one is tube method, the second one is solid phase microplate, and the third one is the gel cut. Okay, after this I will give you a video to show you how we do the antiglobulin test, and also some further readings on antiglobulin test if you find it difficult to understand. If you have any questions, you can ask during the class, or you can ask in the app module itself. So see you then. Bye.